Hi, my name is Paola Nathali Hernández Valenciano. I'm a master's student in Bioengineering and Intelligent Computing at the University of Guadalajara. This poster presents part of my thesis project called Mapping Gene Regulation in Saccharomyces cerevisiae, an analysis of essential gene interactions, which focuses on characterizing physiological states in Saccharomyces cerevisiae and Cleveromyces marxianus through transcriptomic analysis. As part of our introduction, we have that minimal genomes models often assume that gene essentiality is static. However, cells are dynamic and adjust their gene expression depending on internal and environmental conditions. Our objective was to characterize three physiological states: adaptation, viability, and vitality, based on gene expression profiles, and identify key genes that help us define each of these three states. These allow us to better understand cellular requirements in specific contexts and improve strategies for minimal genome design and bioprocess optimization. As part of our methodology, we perform RNA-seq data analysis for both species. We start downloading transcriptomic data of each species and for each physiological states. The experiments that we download the data are shown in figure 1. Once we have this data, we did a pre-processing, quality control of the raw reads, alignment to the reference transcriptome, and then the transcript abundance. We then follow differential expression analysis. We did a pre-processing, data exploration, and finally the differential expression analysis. Once we have the differential expressed genes, we did downstream analysis using them. We filter the genes by physiological states, and then we did a gene interaction network using network analyst. So as part of our results, I can show you this in figure 2 and figure 3. The distribution of these differential expressed genes across three physiological states in Saccharomyces cerevisiae in figure 2 and in figure 3 for Cleveromyces marxianus. So what we can see in these graphs is that in single dots we have the genes that are differentially expressed and that we found uniques for, in this case, vitality, for adaptation, and for viability in Saccharomyces cerevisiae. We also found genes that are shared in the three physiological states, and what we found interesting is that we have genes that are shared between two states, and this may represent a transitory phase from one physiological state to another. This is for Saccharomyces, but for Cleveromyces we have the same info. In single dots we have the genes that are found unique for each physiological state, the genes that were found in the three physiological states, and the transitory phase from one physiological state to another. Then we did a gene interaction network using genes that were shared between the two species, and we found genes that may serve as functional markers of physiological change and key regulators in cellular response. In pink we have genes that are associated with the adaptation state, in green for viability, and in purple for vitality. Finally, our conclusions are that the identification of key genes associated with each physiological state supports a dynamic perspective of gene essentiality. These findings on the delimitation and the characterization of these three physiological states demonstrate that these changes in their gene expression are responding to species-specific features and are reflecting that the cell capacity is dynamic to respond to both physiological and environmental demands. And finally, this perspective has significant implication for the design and optimization of minimal genomes and for the bioprocess. Finally, I want to thank you to see my explanation to my thesis project. If you have any question, you can send me an email and I happily respond to you. Thank you.